My name is Jorge Lannot. I'm from the University of Buenos Aires, Argentina, and I'm here to present my work that's called Enhanced Prediction of Protein-Carbohydrate Complexes through Guided Docking with new AutoDock Vina site. First of all, I wanted to tell you the motives of this work. Protein and carbohydrate interactions are really important in many biological processes. There are a lot of metabolic pathways that are regulated by interaction between proteins and specific carbohydrates. So their study is really important. The main experimental techniques are very important to study these kind of interactions, but at the same time are really limited. The carbohydrates can be really large and flexible molecules, and this makes them difficult to study. So we can see, for example, this effect by searching on the protein data bank of all the available structures that are more than 220,000 structures. Only a bit more of 4,000 are of protein interacting with carbohydrates. So it's a very sub-represented thing in the database, and it goes even lower if you search for high-quality structures and that have big sugars, like made of 4 sugar rings or more. That leads to a bit more of 400 results. It's very little number. So it's a problem that is sub-represented. A main hypothesis used to study this interaction is based on the fact that the crystallographic waters present and the ligand binding site may be giving information about the polar interactions that there happen when the ligand is interacting with the protein. As we can see clearly on this image, the water molecules are exactly positioned where the polar interactions then are done with the hydroxyl groups of the ligand. So in this context, computational methods take a high value to study these kind of interactions where the experimental methods are not able to reach. The main computational method to study protein-ligand interaction is molecular docking. And the most common used program is AutoDock Vina. This program predicts interaction between a given protein and a given ligand, but it's an algorithm trained to use really small, rigid, drug-type molecules. So when you wanted to use it with carbohydrates that are large and flexible molecules with a lot of polar groups, they fail. They are not trained to do this. In this context, there have been a lot of improvements done to this AutoDock Vina. First, the Vina curve introduced a scoring function specific for glycosidic linkages, torsion angles. In this way, the flexibility problem was reduced to only score the glycosidic linkages, and this enhanced the predictions. Then, a few years later, Glycotorch Vina improved a little bit more the results by implementing a scoring function specific for glycosaminoglycans that are sugars with amino groups or sulfate groups. But both of these improvements are not that good. Those are not useful to treat with large carbohydrates, which is a situation that happens a lot on nature. So it's really important, and there are no computational methods available to study these interactions. On my lab, recently, we have developed a new docking tool called AutoDock Vina site. This tool has all the benefits from the other tools that I mentioned, but it also has a new feature. It allows the user to define bias on the docking simulation. So you can enhance some particular interactions with some particular functional groups of the ligands in the defined positions by the user. So having this available over the other improvements, we developed a protocol where we define the biases based on the solvent structure present at the ligand binding site when the ligand is not there. So to see if with that information we could improve the docking predictions. So the objective of this work is to improve the performance of the carbohydrate pose prediction using the solvent structure information to guide the docking simulation through the new Vina site. As we can see here, studying the solvent interactions can improve the ligand pose prediction. So to achieve this, we designed a dataset made of 30 protein carbohydrate complexes with carbohydrates going from 4 rings to 9 rings, so really big ligands, and which had available also their structure without the ligand present. So we were able to study the solvent structure at the ligand binding site. Then we did a docking protocol using the conventional method as AutoDock Vina, VinaCarb, and GlycoTorch Vina, and also with our guided docking protocol based on solvent structure information. Additionally, we developed a positive control guided docking protocol based on the real interaction that we already have, because we have the reference structure, just to study the maximum capacity of a guided docking protocol on docking predictions. Finally, to study the results, we measured the RMSD. That is a measure of how good is the prediction compared to the reference ligand. It's defined that RMSD lower or equal to 2 Armstrong says that the prediction is equal to the reference structure. So in this way, we can see the results on the X axis. We see the percentage of the systems studies that got an first post rank RMSD equal or lower to the RMSD that is displayed on the y-axis. And we can get a lot of conclusions. First of all, we can see that the conventional methods are not good. They only predict correct results in the 30% of the studied systems. Then, on violet here, we can see the results achieved by our guided protocol based on solvent structure. This tells us that studying the solvent structures and getting that information to a guided protocol, a guided docking protocol, can really improve the docking predictions between proteins and carbohydrates. Finally, we can see a high 80% of accuracy of the positive control. That it's very important to see that a correct site definition to guide the docking really ensures good results in prediction, even when you work with larger and big carbohydrates that have a lot of torsions and a lot of interactions. Just to show you one example of the water guided protocol, here I have a reference structure superimposed with the water molecules, and we can see that the waters position almost exactly where the polar functional groups of the ligands then position when the interaction happens. And adding this information to a guided docking protocol really helps to achieve better results than the ones that you get with the best conventional method, that is GlycoTorch Vina. And so it's a method that is not that computationally expensive, but at the same time, really useful to get better predictions. Well, finally, this is my thesis work. I'm a degree student of biology, so I did a lot of work referring to a lot of parts of the study. We studied the we developed a scoring function to specifically rank cluster results. We also studied the effect on bias sites definitions. So what were the limitation and the prospects? And finally, we also studied the effect on the ligands conformation on the docking protocols. But I don't have time to show you all this. So today and tomorrow, I have a poster presentation. And then also Friday, my director, Carlos Moenuti, is giving a flash talk a bit early, but it's going to be interesting because there are a lot of features that can be useful in docking simulation. So thank you very much. There you have an event. Any questions? Or we can talk also at the poster. So
okay,
thank you
so much.